Now let's go over the protocol for the antimicrobial lab. In this lab, we're going to test some various antibiotics on some different bacterial samples, and then we'll also test some disinfectants on the bacterial samples. So to start this lab, you're going to label a large Muller-Hinton 2 plate with the name, the date, and the bacterial strain that you're using. And then you would gently stir your bacterial culture and pipette about 0.75 milliliters of bacteria onto the plate. And then you would use a sterile swab to carefully spread that liquid from the bacterial culture all around the plate. So to really coat the whole plate and streak a lawn on the agar. And once that's all spread out, you would cover it back up and then let the plate dry for 2 minutes. Then you would briefly sterilize your forceps in the incinerator and then let them cool. Once you're sure that they're cool, you would gently place the 5 different antibiotic discs onto the plate as shown in the picture below. So with your sterilized forceps, you have to take a piece of that antibiotic disc paper out and place it on the plate. So you'll do that with the 5 different types of antibiotics, and you'll have the discs laid out on the plate like this when you're finished. And for the disinfectants, you would label 4 TSA plates with your name, date, the bacterial strain, and which disinfectant you will be placing on that particular plate. So we're going to do one with Lysol, one with bleach, one with alcohol, and one with hand sanitizer. And then you would pipette approximately 0.25 mLs of the bacteria onto each plate. And then again, use a sterile swab to spread that liquid around and cover all the agar with that bacteria. So streaking a lawn on that plate with your bacteria. And then again, you're going to cover it and allow the bacteria to dry on the plate for a minute. Then you would use sterilized, cooled forceps, and for each plate, you would gently coat a filter disc with a disinfectant. So these filter discs are just these small blank pieces of filter paper with nothing in them in the paper. And so you're going to be adding the disinfectant, soaking the disinfectant into this paper. So to do this, you pour or spray the disinfectant into an empty mini Petri dish and then dip both sides of the filter disc into the Petri dish with the disinfectant. So you're going to, with the sterilized forceps, just grab one of those blank filter papers and dip it in the disinfectant and really make sure it's coated on both sides. And then place that in the center of the plate of the bacterial lawn that you just streaked. And then you're going to repeat that for all 4 of the plates with a different disinfectant for each disc. And don't flip the plates over because we want to make sure that none of the discs fall off. And finally, you would incubate all those plates at 37 degrees Celsius for 24 to 48 hours.